importance of states, especially in the elections. Clicker's not working, but my computer will work. So. Okay, this one's working. That's great. Okay. Yeah, thanks so much. I'm really excited to be here. I think this workshop has been a lot of fun for me so far. And so I'm excited to tell you about some of my own work in collaboration with Byron Yu, Adam Cohn, and Christian Machens about novel dimensionality reduction technique we've been developing to study interactions between populations of neurons. So just to preface this talk, we're going to be going over the same topic that I covered in the main meeting, for those of you who were there, and I do see several familiar faces. For your sake and for mine, this talk will be slightly different. But also, so with regard to that, this is going to be a more technical talk, and hopefully people will get more value out of it that way. But I'm not going to assume that you went to the main meeting talk, so I've structured this in two parts, which I'm calling passes. So on pass 1, there should be material that's familiar to those of you who are at the main meeting. We'll cover high-level motivation and model description, and then we'll go ahead and directly, once we have an intuitive understanding of the model, show results of applying it to studying interactions between V1 and V2. And then in the second part, we're basically going to start over, but we're going to peel back layers of complexity, and we'll dig into some math. So hopefully that will make people more satisfied. So starting with this first part, I'm interested in the question of how brain areas interact. I think answering this question is critical to understanding virtually all the computations that the brain performs. And in particular, I think this is the most exciting time to be asking this kind of question. I think, especially at the main meeting this year, it's been thoroughly demonstrated that technological advances have made simultaneous multi-area recordings increasingly common. But while these promise to reveal a lot about how brain areas communicate with each other, it's that answering that question of how they interact is still a challenging statistical problem. These population signals are high dimensional, dynamic, and inherently variable. But a widely adopted approach now to addressing some of these challenges is dimensionality reduction. Now, of course, in yesterday's session, we covered some fairly advanced dimensionality reduction techniques, but just to address it at a high level, dimensionality reduction takes population activity and extracts a smaller set of latent variables, and these latent variables capture prominent activity patterns that are shared among neurons. But current dimensionality reduction techniques are ultimately inadequate for asking or answering how brain areas communicate with each other. And to see why, I like to take a step back. So if we consider Area A relays certain activity patterns downstream to Area B. And so these relayed signals, or I should say Area A will reflect patterns that are shared among neurons across these areas. But not all of Area A's ongoing computations are relevant to Area B. And so its activity will also reflect patterns shared among neurons within Area A, but not with Area B. But of course, this story gets more complicated when we recognize that Area B has a lot to say to Area A. So communication is bidirectional, and not just bidirectional, but asymmetric, and occurring at multiple time delays. And these time delays probably come about due to various anatomical or computational considerations. And so if population activity reflects both relayed and unrelayed signals, then so will our extracted latent variables. And so this is the fundamental problem with current techniques when it comes to studying inter-area interaction. In particular, it leaves two critically unanswered questions. First, how do we distinguish population signals that are relayed across brain areas from those that are not? And among the relayed signals, how do we distinguish feed-forward from feedback? And so these questions are the ones that we sought to address with our dimensionality reduction technique called DLAG, which stands for delayed latents across groups. And here's the high-level idea behind the DLAG model. We start with population activity in each area, and we decompose it into two sets of latent variables. A within area set, which captures covariance between neurons within a population, within an area, and then an across area set, which captures covariance among neurons across areas. So we model the time courses of these latent variables with Gaussian processes. I'll be talking about that quite a bit more in the second part of this talk. But for now, all that you need to know is that each of these latent variables is associated with a characteristic time scale, which tells you how quickly they vary. But critically, for this model, we also associate each across area latent variable with a relative delay. And so if a particular time course is reflected in the population activity of area A, and a similar time course is reflected in the activity in area B, but after a time delay, this model will capture that phenomenon. So we can also capture multiple time delays, and based on the sign of these delays, we can start to assign some sort of directionality to these across area latents. So I'll go into, in the second part, the observation and state models for this, for DLAG. But for now, all we need to know is that we can learn the observation and state model parameters, including these delays, from data. And then once we have those parameters, we can infer these latent time courses on a single trial basis. So hopefully that's enough for you to have a flavor of the model. And now I'm going to go ahead and start showing you how we can apply it and interpret its outputs on real data, studying interactions between V1 and V2. So first off, V1 and V2 are a great place to study inter-area interaction, and also a great launching pad for methods like this. We know anatomically that V1 and V2 share strong reciprocal connections, and so they strongly interact with each other. And so with that in mind, we recorded from about 100 neurons in V1 with Utah arrays, 25 neurons in V2 with tetrodes in an anesthetized macaque, while he passively viewed an oriented grading stimulus. We separately analyzed 8 of these orientations across 2 different sessions for a total of 16 data sets. And in order to prioritize the more technical discussion in part 2, I'm only going to provide highlights of this analysis. But I want to emphasize that the trends that I'm going to illustrate were consistent across all of these data sets. So we applied the DLAG model to spike counts counted in 20 millisecond time bins. And now what I'm going to show you are the latent variables and time courses deemed or extracted from the data and deemed significant using cross-validation. So let's start with the patterns that are shared among neurons across areas with multiple delays. So here what I'm showing are 2 latent variables and their time courses identified with very different time delays. So we have single trial traces in gray and trial average in black. So just starting at the top, we have a identified delay of +5 milliseconds. And so based on the sign of that delay, we might call this a feed-forward signal from V1 to V2. And what immediately stands out in this feed-forward activity is just how strongly it reflects that sinusoidal drift from the grading stimulus at just above 6 Hertz frequency. Then going down below, we have this identified time delay of -27 milliseconds. And based on that sign, we might call it feedback. And what we can see that this feedback activity ramps up much more slowly and has this broader peak. But then what was especially striking in this data with regards to these features was just how consistent they were across data sets. So what I'm now representing here is I've taken these latent across area signals. I'm representing each one as an ordered pair, where we have their Gaussian process timescale on the vertical axis and their delay on the horizontal axis. And so these latent variables were extracted on, from responses to a single orientation. But across all orientations, for this example session, we found remarkable clustering around these points. So now let's turn our attention to the patterns that are shared among neurons within an area, but not across areas. So again, these were deemed significant using cross-validation. And just the fact that we find these exclusively within area trajectories suggests something pretty interesting, which is that we have found substantial activity that's shared among neurons within an area, but not shared with neurons across areas. So more simply put, not all activity is relayed between V1 and V2. So now let's bring in our across area latents to take in the whole picture. So if I were to interpret these results, I actually think these are fairly consistent with my intuition for hierarchical processing in the visual system. Namely, stimulus drive tends to be bottom up. And then as we get further up the hierarchy, in this case to V2, the relationship to the stimulus is less straightforward. Of course, it would be pretty interesting to see in future work what these traces actually are used for, or this activity is actually used for in the brain. But at this point, this concludes our first pass. And hopefully you have a general idea of what constitutes the model, as well as how we might apply it and interpret its outputs when we apply it to simultaneous multi-area recordings. But now in our second pass, I'm basically going to start over, but I'm going to take you through our thought process in designing this method. And so I'm going to start by reminding you of our key statistical questions, which was how do we distinguish the population signals that are related across brain areas from those that are not? And among these related signals, how do we distinguish feed-forward from feedback? So when we started asking these questions, the DLAG model didn't exist yet. And so we first considered what kinds of dimensionality reduction techniques can we use to get at these challenges. So the first class of techniques, and I've already given away the punchline for this slide, but we might think of principal component analysis, factor analysis, Gaussian process factor analysis, and the many extensions. All of these methods, to some extent, they're trying to capture prominent activity patterns that are shared among neurons. And as I mentioned, the problem is these, without additional structure, we can't distinguish signals that are related across areas from those that are not. And so ultimately they're inappropriate for multi-area recordings. Now, there is of course a dimensionality reduction technique that is very appropriate for multi-area recordings, and that's canonical correlation analysis, or CCA. Partial least squares or reduced rank regression are also part of this family. But at a high level, CCA takes two populations, and from those observations, it extracts a set of across area latents that optimally capture the correlation in the activity between the two areas. And then it throws away all the remaining activity away as noise. So right off the bat, that might be a problem if you care about those within area signals. But even if you don't, first, there are a couple issues with the across area side. CCA has no concept of time, which I've tried to illustrate with these more jagged time courses. And in addition to that, that just makes it that much harder to try and tease apart feed-forward from feedback. And so this motivated our choice of, our modeling choices for the DLAG model, having these within and across area delayed latent variables. So now I'm going to start trying to formalize the DLAG model. This has been a very intuitive description. And so to start getting there, let's recast this as a graphical model. So now we have our observed activity in each area, cast in these vectors y vectors. And then each area is influenced by two sets of latent variables, a within area set and an across area set, which actually influences both areas. But what I'm trying to illustrate here is that this across area set influences area 2 with some sign delay. And then we string together these latent processes, these latent states, with a Gaussian process. So now I'm going to dive into the state model and observation model. And let's start with the state model, which tells us our assumptions about how these latent states evolve over time. So first, let's start with the within area state model. Here we model each within area latent signal as being drawn from an independent Gaussian process. So these are zero mean Gaussian processes with covariance kernel k. And in Anji's talk yesterday, we did discuss a little bit. You can choose any number of kernels for this Gaussian process, but for this application, let's just stick with the squared exponential kernel. And so the main feature here is this exponential term, which just says that 2 time points, the covariance between 2 time points decays exponentially as they become farther apart. So this should look very familiar to people who are already familiar with Gaussian process factor analysis, GPFA. But now let's move over to the across area state model. So still here, the across area latent states are modeled with independent Gaussian processes, and still with a squared exponential kernel. But now we snuck in this delay into the delta t term. So I'm going to pictorially illustrate what this actually means, but I'll just highlight a couple advantages of this approach. First, by defining the delays in this way, we're defining them continuously. So they're not tied to your choice of spike count bin width. And then as a direct consequence, we can actually directly optimize these with gradient updates. So in the end, this whole model is trained with exact expectation maximization, maximum likelihood framework. And then another aside is we've chosen to model the time courses with Gaussian processes. We didn't need to do that. For example, we could have, in principle, modeled this time course with a dynamical system. But among other considerations, this advantage in particular drew us to Gaussian process. So now let's talk about what this delay actually means in this Gaussian process. So really what's happening is we assume that both areas have access to the same Gauss underlying process. But area A accesses this process within a certain window. Area B also has access to that process, but in a window that's shifted relative to area A. And so that's incorporated with this delay D. So overall, the across area time courses are just shifted views of the same Gaussian process. So this tells us something about the temporal evolution of the system. Then the observation model is going to tell us about the structure of our observations. And so now I'm going to throw away the concept of time for a moment, and I'm going to move to this population state space view. So here we have, we're representing population activity with this neural state space, where each axis, of course this is high dimensional, but I'm only showing 3 axes for illustration. So each axis represents the firing rate of an individual neuron. And so now here's the observation model. We define the activity of a given population as a linear combination of these within and across area signals. And so this has a really nice interpretation that, especially for these matrices CW and CA, which is that just starting with CW, the columns of this matrix actually define a subspace that lives within this population space. And so now we can interpret this within area A latent space as being basically any activity that lies within this subspace, captures the covariance that's shared among neurons within that area, but not with area B. And so analogous on the side of area B. And then looking at the CA matrix, the columns of CA also define a subspace. And this subspace, any activity that lies within this subspace, within area A, is highly related to activity that lies within the analogous subspace in area B. But we don't have to stop there, because the state model with the time delays allows us to partition these across area signals into delayed components. And so what we can actually get is additionally partitioned subspaces where, take this one for example, any activity that lies along this subspace captures is correlated with a corresponding activity in this subspace, but with a positive time lag, for example. And so altogether, this DLAG model actually gives us three subspaces, if you will: a within area, a feed forward, and a feedback subspace. And I actually interpret, or I tend to think about the real data that I've shown in part one with this picture in mind. But I think I'll leave it at that.
Are these subspaces all necessarily orthogonal?
There's no constraints of orthogonality, but they are going to be linearly independent. Yeah. Okay, so with the last few minutes that I have, I'm going to touch on how we validated this model on synthetic data. I don't want to go into the details because I really wanted to prioritize this discussion, so I'll just give you a flavor. So DLAG is not just a tool for data-driven learning and inference, but it's also a generative model. And so from that, we can actually generate synthetic data where the ground truth is known. And so toward that end, here, as an example here, we could generate within and across area subspaces with dimensionality shown in parentheses. And then from that, we can generate population firing rates that actually matched our neurophysiological data from part one. Matched in terms of number of neurons, number of trials, trial length, and population statistics. And so, like I did in the main meeting, I'll highlight just two aspects of DLAG's performance as they are relevant to interpreting the real data from part one. So first is, here we have represented within and across area time courses on a single trial. Across many trials and simulations, DLAG accurately recovers these single trial time courses. And so consequently, also these within and across area subspaces. And then here what I'll show, again, just to demonstrate that DLAG recovers these delays very accurately. So we saw this in the real data portion, where I'm representing the across area signal as an ordered pair with its Gaussian process timescale versus its delay. And so in this case, we had a 10-dimensional across area subspace. And so we had 10 such across area delay timescale pairs. And you can see that the estimates very closely match the ground truth. So this highlights a point I made earlier, which is that because we model the delays continuously and time continuously, even though in these simulations the model only had access to observations in 20 millisecond time steps, just like our 20 millisecond spike count bin width, we were able to recover delays with 5 millisecond resolution. So we can recover delays with sub bin width resolution. And so there are a number of other ways that we validated this DLAG model. So feel free to ask me about any of these, but I think I'll go ahead and end it there and take any questions. Yeah.
In your example, on synthetic data, I was curious how you can recover, how robustly you can recover the dimensionality of those subspaces within an ecosystem.
Yeah, so this is bullet point number 3 buried on this slide, which is the way we recover the number of latents is with cross-validation. And I don't have a slide to show you this, but we are consistently able to recover the correct dimensionality for each subspace using cross-validated log likelihood as our metric. Yeah?
How will you scale this up to more regions with only sources, for example?
Yeah, so the question, if people couldn't hear, and if I'm understanding, how do you scale this to maybe more than 2 brain areas, right, to multiple brain areas? And the answer is, you can do it. And let me just pull up the graphical model so we have something to look at. Actually, built into this framework, I've defined this pretty generally, so that actually I've already defined this for multiple brain areas. But then the question is about scaling. And there are 2 issues with scaling. So the first one is, the easiest thing would be maybe for each brain area, we could have assigned their own individual within area latents. And then we could just have one set of across area variables for all of these brain areas. So the problem there is, in terms of scaling, is scaling of interpretability or interpretation. This might be too technical, but I'll go ahead and just say it, that if you had such a scheme, you would not be able to tell if a latent signal was shared across all areas, shared among only one pair, a triplet, and so on. So that's one issue with scaling. The other issue with scaling is if you want to get around that issue of being able to distinguish if an interaction is pairwise versus shared with the whole group of brain areas, then you would need to include latent variables for each pairwise interaction or triplative interaction, etc. And so then you have an issue of scaling with cross-validating hyperparameters. So for now, I tend to think of this method as being most useful when you know two brain areas are interacting and you'd like to know more details about the actual computation going on between those two areas.
I was just wondering, I guess back to the Sumedo paper.
Mm-hmm.
So what's nice about this, I mean, in addition to the delays, is that you're able to use data from both areas to extract latents versus seeing them in one area and then seeing how they, have you done across have you compared those sorts of methods? Because especially with multi-brain area thing, I can imagine that using that technique sometimes might be
so the question is, have we compared, say, predictive performance between like the reduced rank regression and others?
So,
or?
yeah, you can just extract latents from one area and then see which of the, you could, so you could just take V1, 100 units, extract all the latents, and then see which ones match in V2 with sufficient delays using a GLM. And I imagine that won't be nearly as powerful as this technique. But
Right, I haven't done,
pretty well.
yeah, I haven't done that exact comparison, but we've been thinking about how this compares to other models, and I have reason to believe that this will outperform. You had a question over here?
Yeah, I was curious, going back to interpretation, if you ever try to generate data from a spiking model, let's say you have two circuits as they synchronize and there is a variety of these models available, what kind of latency, let's say this cross area synchronization would allow?
Yeah, I can't directly answer that, but I can tell you that we have been thinking about, so here I presented synthetic data results for the case where we have our synthetic data matched our model assumptions. And so then the question is, how do we do once we break those model assumptions? So down here, I highlighted that we have started to apply this to model mismatch scenarios. Take, for example, one example is you could just take this, I skipped it here, this generative model framework, and then add Poisson variability at the end, for example. We still seem to do okay in that case. The other scenario is you could imagine that maybe we generated this data from a recurrent network as the ground truth. And I can talk to you more about that offline, but the short answer is it gives reasonable output. Yeah, but I can tell you more precisely what I mean maybe later. Yeah?
Just out of curiosity, have you tried this on non-anesthetized data sets where you would expect the structure to look that much different?
That's what, so I can answer that in two parts. The first answer is I'll have a good answer for you soon, because we're currently analyzing data from awake behaving animals. The second answer is we have, in some of our previous work, we have looked at simultaneous recordings in both anesthetized and awake monkeys. And we've applied more basic methods like CCA, factor analysis, and those have actually returned qualitatively similar structures in both the awake and anesthetized.
If there are no other questions, let's thank everyone one more time.
Thank you.
And our next speaker is Tatiana Engel.